Hey, my name is Peter Issa. I'm a first-year medical student here at LSUHSC in New Orleans. Foremost, I want to thank Dr. Fern Chen and her team for allowing this opportunity, and I want to go ahead and thank the judges as well for making this opportunity possible. So over the summer, I had the opportunity to join the CNRC Research Scholar Program. And so there with the Clinical Neuroscience Research Center, I was able to review the repurposing of ATN161. And so this is our work. Our title is The Potential Role of ATN161 as an Integrin alpha-5 beta-1 inhibitor in preventing common viral infections. This is a view of our total poster. Now, integrins are transmembrane proteins, and so they sit on the cellular membrane. And so in this way, they have many functions that are not limited to immunological function, cell adhesion, and apoptosis. Now, cell adhesion is actually particularly important because viruses and bacteria can use this cell surface protein, correct, and internalize and actually get into the cell. And so the one specific class of integrins are the RGD integrins. And so these are integrins that contain the sequence arginine, glycine, and aspartate. And so these ones in specific have been shown to be implicated in the adhesion and the internalization of viruses. And so over here, you can see our figure 1 on the right. We can see that integrins are actually dimers. And so here's our alpha subunit, and this is our beta subunit. And in specific, one RGD integrin is alpha-5 beta-1. And this specific integrin is actually very important and has been known to mediate many viruses entering cells. And so the literature has shown that, and this is the purpose of our review. Now, ATN161 right here, as you can see on the right, is a pentapeptide, and it's been shown to be a non-competitive inhibitor of this alpha-5 beta-1 integrin complex. And so this ATN161 was shown to be an anti-tumor agent in a 2006 phase 1 clinical trial. And so it was shown to be safe there, and it showed a U-dose response curve. And so in this way, we're looking at its repurposing as a potential antiviral therapeutic. Now, what we did was we went to PubMed and entered the searches as well as into Google Scholar. And we were able to find a total of 14 papers that had looked at this within the last 3 or 4 decades. Now, we were not able to include, or we made sure to exclude, any studies that looked at other integrin complexes. So it had to be specifically alpha-5 beta-1, because that is the integrin complex that ATN161 is able to non-competitively inhibit. So here is the data in whole, and you can see that ATN161, integrin alpha-5 beta-1 is mediated in 7 different viruses entering cells. And so you can see them here: human adenovirus type 2, Epstein-Barr, HIV, foot and mouth disease, Ebola, PHEV, and actually the new SARS-CoV-2 virus, which is actually causing this pandemic. And so you can see that integrin alpha-5 beta-1 is implicated in all of these. And as I said earlier, it mediates either cell attachment or cell entry because it's a transmembrane protein. And so I encourage you to take a look at these references if you care to read a little bit further on. What I would like to highlight is those studies would actually look at ATN161 in a murine model. And so that was only these 2 studies here at the bottom, PHEV and SARS-CoV-2. And so what they found with the PHEV study, Liv et al. Found this, and they found that, and these figures correspond actually to the real data. And so they found that PHEV infection was able to roughly double the count of alpha-5 beta-1 integrin. And so that means that when a cell is infected, were ATN161 to actually inhibit infection or attenuate infection, that this could be a targeted approach, because in the virus increasing the integrin count allows a targeted approach. And so you can see that treatment with ATN161 lowers the integrin count to 1/4 of what it was, and then simultaneous infection and treatment lead to half the viral load. And so that's very important. We can see that ATN161 is able to attenuate PHEV infection. Now, moving forward to SARS-CoV-2, and this is really a very interesting finding here, is that again, SARS-CoV-2 infection induces roughly double or twice the amount of integrin alpha-5 beta-1. So again, this can be a targeted antiviral therapeutic. Now, simultaneous infection with ATN161 treatment, we have roughly the same number of integrins, the alpha-5 beta-1 integrin complex, and then half the viral load. And so once again, we're able to see that ATN161 really was able to show, at least in these two viruses, SARS-CoV-2 and PHEV, that it was able to decrease the viral load by half. Now, our findings here I've already summarized pretty neatly, but they're here in words. But I do want to emphasize once again that many viruses upregulate their expression of alpha-5 and or beta-1 integrins. And so that allows for a targeted approach. And that means that ATN161 could really be repurposed and could be expedited through and pushed through clinical trials sooner, hopefully sooner than later, to be a potential therapeutic against alpha-5 beta-1 integrin mediated diseases, specifically SARS-CoV-2. And so to bring the discussion full round, we know that integrin, specifically RGD integrin, mediate the pathogenesis of many viruses and bacteria even actually. So we were able to summarize 7 viruses that follow this alpha-5 beta-1 integrin. And we were able to show that viruses upregulate the expression of alpha-5 beta-1 integrin, and that viruses actually downregulate the alpha-5 beta-1 integrin complex. And so we want to, of note, state that although integrins mediate many functions, many intracellular functions especially, and many signaling pathways, FDA approved medications, integrin targeting medications have been approved, such as one against, as an antagonist against alpha-4 beta-1, and there's another one against alpha-2b beta-3. And so these alleviate multiple sclerosis and Crohn's disease, as well as acute coronary syndrome, respectively. And so this really highlights the potential for ATN161, which is an alpha-5 beta-1 integrin antagonist, to really prevent and treat alpha-5 beta-1 mediated viruses, with a particular emphasis on SARS-CoV-2. Here's one more look at the poster in large. And of course, thank you. I went ahead and attached my email here. Please reach out. Please do not hesitate. Thank you.